Okay, in this topic, we're actually moving into chapter 9, and it will introduce you to protein synthesis. Okay, here's the overview of protein synthesis. Once again, now that you know a little bit about DNA, you know a little bit about RNA. Of course, you learned about proteins previously. Then hopefully we can put this all together. And again, remember that this process is totally different than DNA replication, which we talked about previously. DNA replication takes place in the nucleus of a cell during the S phase of interphase in preparation of mitosis. Okay, this protein synthesis is how we actually get from a code that's in our DNA to proteins that actually function in some way for the cell. So again, what we're going to do is start out with very generalized information, go a little bit deeper into more detail, and then come back out so that you understand the whole picture, at least in a very general fashion. Okay, so this is a nice summary. DNA provides the code for RNA to come in and copy that code, so to speak. Then that RNA is going to go out of the cell and eventually we get to a protein. This first part is called transcription, where we go from DNA to RNA. The second step, RNA to protein, is called translation. Again, seems very simple. You don't know any details about it yet, but this is a lot of information right here. And as we get into more detail, hopefully you'll understand it a little bit better. Okay, here's a nice overview from an old textbook, rather simplified, but that is okay. This area right here is supposed to be the nucleus of a cell. This area out here is the cytoplasm, and this is a ribosome that is out there in that cytoplasm. The process of protein synthesis begins when DNA opens up in the nucleus of the cell, and there's a process that we call transcription in which a strip of messenger RNA is produced. And that strip of messenger RNA comes directly, specifically by complementary base pairing from that DNA code. Then that messenger RNA leaves the nucleus, goes out through a nuclear pore, sort of strings through the ribosome, and from this area, we have transfer RNAs, not shown in this overview, but those are going to bring these little amino acids, shown here as little dots, little round things. They connect, and that is our protein. So this first step is transcription, takes place in the nucleus. The second step, translation, takes place out here in the cytoplasm of the cell at the site of the ribosome. This is the way your book shows this. It doesn't have all the extra material, the extra drawings of where are we in the nucleus, all of this kind of thing, but it's sort of a stylized drawing that does cover the process. Again, transcription is where we go from the DNA to the messenger RNA. And look carefully at these different letters. What's happening here is when this DNA opens up in the nucleus of the cell, and this we're going to call one gene, one certain area of this DNA. These would be down here in brown, the various nucleic acids of RNA that would attach. So where there's this T right here, of course, it would be the A. Excuse me, let me back up. Where there would be a T right here, that doesn't even make any sense. Where there's T right here, what would attach would be this A. I should have made that like right there. Okay, where this A is exposed, you might first think, oh, a T, oh, a T. No, because we're talking about messenger RNA, there is no thymine. So the one that would come in and attach right here would be the uracil. Okay, and so on. And again, that process is called transcription. This would take place in the nucleus of the cell. Okay, then we have the messenger RNA. It's not showing all the detail, but this strip of messenger RNA would leave the nucleus, go down into the cytoplasm at the site of the ribosome, and again, it's leaving out the step of the transfer RNA, which we will discuss in just a minute. But by means of that transfer RNA, we will end up with a series of amino acids, which, although pretty tiny protein, we're calling it a protein. Okay, so now we're going to look at it in a little more detail, step by step. We'll start here with transcription. Now, one thing I want to mention here that may help you get these straight, that transcription is first and what's going on here. Think about what transcription in general means, not necessarily in biology. Transcription is when, for example, someone takes the written notes, or wait, not the written notes, maybe an oral recording and puts them into the typed word, okay, a written hard copy. That's transcription. You're still going from, for example, English to English. There's no huge change. It's just the form is changing. The same way when you go to DNA to RNA. They are both nucleic acids, so you're staying in the same language. So that's what we call transcription. While I'm here, I'll go ahead and explain translation, even though we'll do that on the next page as well. But RNA to protein, if you translate something in the regular use of the word, not in biology, translation might be going from English, say, to French. Okay, you're changing languages. When you go from RNA to a protein, these are totally different structurally. This one is a nucleic acid. This one right here is a different class of biological compounds. Proteins are totally different. So in this case, that would be translation. Another easy way to remember which one's first and which one's second, kind of stupid, but the beginning of these two words, transcription and translation, are pretty much alike. Exactly, trans. The first letter that's different here is a C. The first letter that's different here is an L. And of course, C comes before L in the alphabet. So transcription is first, translation is second. Okay, let's explain what happens here. For transcription, a portion of the DNA, which is there in the nucleus, unwinds. And we're going to call that one gene, one stretch of DNA that's going to code for a particular protein. The RNA nucleotides, which we will call codons, and I'll explain this again later, these are going to come along. There are free nucleotides in there. They're going to pair with the exposed DNA bases. The bases are called the code on DNA. Again, we'll review that in just a minute. But this is that complementary base pairing. The only difference here is that RNA does not have thymine. It is replaced by uracil. So the end result of this is a long strip of RNA, which we call messenger RNA. Then the DNA closes up. This DNA can be used repeatedly throughout the life of that cell for providing the template for this messenger RNA. We'll move on to translation. As I said here, rewrite in a different language because we are going from RNA to a protein, two different classes of biological compounds. That messenger RNA that we just produced in step 1 is going to now leave the nucleus through a nuclear pore, threads through a ribosome out in the cytoplasm. This is where the transfer RNA comes in. Remember, the messenger is a messenger. It travels. It goes from nucleus to cytoplasm. The transfer RNA, on the other hand, is a short little thing and only has 3 exposed bases on it, and that's called the anticodon. And what this does is match up with complementary base pairing to the exposed messenger RNA codons. And of course, what is it transferring? Amino acids. It has those amino acids attached to it. So once it arrives at the ribosome, the transfer RNA hooks up with the messenger RNA, then moves away, leaving behind the amino acid. After a series of transfer RNAs have come to that site and left behind their amino acids, those amino acids join to form a polypeptide, which is just another word for a chain of amino acids, which in this case we're calling a protein. A protein, of course, as you know, is usually quite large, has several chains of amino acids involved in it, but we're just sort of simplifying it and we're calling a few amino acids, we're calling it a protein for this lecture. So we're going to look at this in several different views. Again, I encourage you to Google protein synthesis and you'll find all kinds of information and images that may help you understand the process. Okay, so we'll start off with this one. This is actually from a textbook published by Benjamin Cummings. And this blue area is supposed to be the nucleus of the cell. The DNA has the phosphates and sugars over here in sort of a light yellow or orange color. And we can tell that that's DNA because it has thymine in it. Okay, then the codons of the messenger RNAs will match up until you have that long stretch of messenger RNA. That is going to leave through a nuclear pore. We don't see the ribosome shown on here, but this would be at the site of the ribosome. And the messenger RNA, as it goes through that ribosome, the transfer RNAs arrive carrying specific amino acids. Again, and I have this on a chart later, so if you're not understanding this fully, don't worry. DNA, it's called the code. Messenger RNA, it's called the codon. And then transfer RNA, it's called the anticodon. Okay, and again, the final image here is of the messenger RNA, the transfer RNA coming down, attaching and leaving, and end result is our protein. Protein synthesis. The step in here, transcription. The step down here is going to be translation. Okay, we can even look at it in a little more detail if you would like to. Once again, the gene is a certain portion of the DNA. Here it is shown. Now we see it opening up and the messenger RNA coming in and doing the base pairing, complementary base pairing, until this nice strip of messenger RNA is produced. Every 3 nucleic acids, every 3 nitrogenous bases consist of 1 codon. Okay, it also shows, which I hadn't mentioned before, RNA polymerase. Don't worry about that too much, but that is an enzyme that helps the RNA polymerase, the RNAs, the messenger RNAs attach. Okay, now we're moving on, and that was transcription. We're moving on to translation. Once again, they come along just as in the previous, a couple of slides before, and we have the transfer RNAs carrying the specific amino acid coming in, complementary base pairing takes place at the site of the ribosome, and as more and more go through, we move on to the next. Is it there? Yeah, I skipped one. Okay, here it is. I hit it twice, I guess. So anyway, as they do this, they will move off and be reused. The transfer RNAs will pick up more of the specific amino acids they can carry. And then as they do this, bonds form between those amino acids until eventually you have the protein. Oops, it's gone. What we're going to do now is draw this. And there is also an assignment for you where you do basically the same thing, only I have given you different letters, different DNA nucleotides, so that you can make sure you understand the complementary base pairing. Okay, so you'll probably need about a whole page, so, take your time on it. But we're going to start out, I think I've left you enough room in your notes. This little thing right here is just supposed to represent that is the nucleus up there. Okay, and I've labeled it nucleus. Here's the DNA. We have our double helix. This is an area that we're going to label the gene that has, and I totally chose these randomly, the 4 different options of the nucleotides that are on there. So that's how we're going to start this diagram. Okay, now I did label on here that because this is DNA, this is called the code. The messenger RNA is going to have the codon. And what I did is helped you with the first three and a half, let's call the 3.5 nucleotides that would have matched up with those exposed DNA codes. The G with the C, the A, you might think T, but no, it's RNA, so it has to be a U. The C with the G, and go ahead and fill out the rest of those. As you're doing this, I'm going to go through it pretty quickly. You may have to, change stop this video if you want to so that you can do it, or watch the whole thing through first before you try to draw it. Okay, hopefully you got these letters. Okay, so these are the ones that would match up and with complementary base pairing from the DNA to the messenger RNA. Now we have the messenger RNA leaving the nucleus through a nuclear pore and going out here into the cytoplasm. This and this messenger RNA in here and this messenger RNA down here are identical. They are the same structure. I'm just trying to show that it moves out. And so here I've labeled the cytoplasm. Here I've labeled this as messenger RNA, and I've drawn on one ugly ribosome. Okay, remember that it's actually 2 subunits, and they it's sort of understood that they kind of, the strip of messenger RNA sort of runs through it, and that is going to be the site where the transfer RNA will come in. Okay, you see I did some kind of fancy stuff. First I went in and put the letters in, and I went, no. I should let the students try to figure out what the complementary base pairing should be. So what I've added are transfer RNAs. Here they are. Anticodons are the letters that would associate with these. So again, now what you want to do is try to fill in the matching letters. Okay, hopefully you got them all correct. Here they are, GAC, UCU, GAU. The next thing you see on this one is that the amino acids that the transfer RNAs were carrying are now connecting to each other, shown by those little dashed lines. And our final drawing here, here we have the amino acids together, and that's what we're calling a protein. And then finally, just remember that the first step that takes place inside the nucleus is going to be transcription. Translation takes place out in the cytoplasm. Okay, here's a question for you. Which of the following best describes protein synthesis? A, sister chromatids are produced so that the identical DNA code can be passed on to other cells as they undergo mitosis. Great description, But that description is not of protein synthesis. That is a great description of DNA replication. Okay, B. Half of a DNA molecule temporarily leaves the nucleus to provide a template for amino acids. A lot of things wrong with that one. First of all, DNA molecule never leaves the nucleus, and there is no template for amino acids. So again, not it. C, a portion of a DNA molecule in the nucleus provides a template for messenger RNA, which goes to a ribosome in the cytoplasm, where transfer RNAs arrive carrying specific amino acids which form proteins. Very long, but correct. D, transcription follows translation in the process of protein synthesis. I wanted to put this one in here because I want to make sure that you realize you always have to read carefully. At a glance, you might say, yeah, transcription's first, then translation. That should be right. But that's not what it says. It says transcription follows translation. So again, that would be incorrect. C is the correct answer. Okay, so now we've gotten into it in pretty much detail. You've redrawn it. And one of the things that I want you to remember about this process is that just like anything else you learn, it's a story. There's always a beginning, a middle, and end. There's always, once you know what happens and understand what happens, it's just like any other story, Hansel and Gretel, whatever. You've just got to keep things in perspective and make sure that you understand the process and the order that things occur. Here's another diagram. Here's the nucleus over here. And we have our gene right here. Okay, that's the little stretch of DNA that has opened up to expose the bases. RNA comes in to form the messenger RNA. It leaves through a nuclear pore and threads through the two subunits shown right here in pink of the ribosome. Then along come the transfer RNAs, which are transferring amino acids, very specific amino acids. They only have the 3 exposed bases, which is called the anticodon. They come in, attach by the complementary base pairing. Then they move away, leaving behind these little colored balls here, which of course are representing the amino acids. When they're all in a chain together, we call that a polypeptide chain, or for our purposes, that's the protein. Again, transcription takes place inside the nucleus, translation out here at the ribosome in the cytoplasm. One more time. Again, another artist's rendition of the same process. We have the DNA opening up. We have the messenger RNA forming here in the nucleus, which is all transcription. Leaves the nuclear pore, comes around and threads through the ribosome, which is right here. Then this codon of the messenger RNA interacts with the anticodon of the transfer RNA, comes along, attaches, moves away, and then you have a growing chain of amino acids which forms your protein, which is your final product. Again, transcription here, translation out here. This is the final picture and the last slide for this topic. Again, this is one from your textbook, has some very nice colors in it, and again doesn't have all the detail, but transcription takes place here in the nucleus, and you can see that a portion is opened up, what we're calling a gene. Messenger RNAs match up with that. So it's code to codon of messenger RNA. It's going to leave through a nuclear pore, thread through the 2 components or subunits of the ribosome. It doesn't actually show this transfer RNA interacting here, but it would, and it would carry along the amino acids, which are shown here as these green balls. Okay, so here we have our chain of amino acids. That's going to be a polypeptide, or for our purposes, that's the protein. That's what we're trying to make. That's the deal. And the other thing you can see here is that this same strand of messenger RNA is going through another ribosome here, another one here, another one here. And in this way, messenger RNAs can produce more protein. So the more ribosomes there are, the more protein they can produce. And you may remember in our first, when we were studying the cell, that sometimes you have ribosomes separately out in the cytoplasm. Sometimes you have groups of ribosomes strung together, which we call a polyribosome. And then don't forget about the rough endoplasmic reticulum, where you have they're peppered with ribosomes. And so once again, this ties back with the idea that cells that have a lot of rough endoplasmic reticulum, because of all those ribosomes, those are the ones that are going to have that are going to produce the most protein. So again, this is an overview of protein synthesis, and in our next topic we'll go over a few more details on this, but basically this is it. This is the story.